Axis formation. Cell fates are specified by cell-cell signaling and the asymmetric distribution patterning of the transcription factor. These TFs activate or repress transcription of specific genes that acquires them. This asymmetrical distribution pattern of the molecules begin during cleavage, because as we said before, the cleavage distribute this transcription factor these distribute along the blastomeres. So this asymmetric distribution of patterning molecule begin during cleavage and generally follow one of the three mechanisms. The molecules are bound to the egg cytoskeleton and is passively acquired by the cell to obtain the cytoplasm. The molecules are actively transported along cytoskeleton to each particular cell. The molecules become associated with a specific centrosome and follow that centrosome into the two mitotic sister cells. So basically, this is the three methods of how these morphogenetic molecules or patterning molecule or transcription factors are recruited into the developing blastomere during the process of cell division. Once this asymmetry has been established, one cell can specify a neighboring cell by paracrine interaction. So embryo, by the end, for the axis formation had developed into three crucial axes: anterior-posterior axis, dorsoventral axis, and right-left axis. And you can see here from the figure how these axes are illustrated on the figure. Four invertebrate groups, sea urchin, snail, ascidians, and C. elegans, have been important model system for developmental biologists. Why? Because they easily maintained in the laboratory and represent a wide variety of cleavage types, pattern of gastrulation, and ways of specifying axis and cell fate. Despite their differences, the embryo of these four invertebrate groups are categorized as type 1 embryogenesis. Why type 1? Because there is immediate activation of zygotic genes, there are rapid specification of blastomeres, and the number of cells are few at the start of gastrulation. Early development in amphibian axis formation. Amphibian embryos, including frog and salamander, is the organism of choice for experimental embryology because they have large cells and have rapid development. If you look to the egg of the amphibian, the characteristic its type is mesolysisal egg. So you see that yolk is concentrated in the vegetal hemispheres, and this delays cleavage. Thus, the first cleavage begins at the animal pole, slowly extend down into vegetal ball. So you can see that the molecule that specifies the dorsal fate are located ventrically, the one with the light blue. The sperm enters from the left side, as you can see, and the cytoplasm at the edge of the cell is called cortical cytoplasm. Once there is a fertilization, as we said before, there is a cortical rotation. This cortical rotation leads to the merging of the cortical cytoplasm of the ventral part with the cortical cytoplasm of the animal part. So the merge leads to the movement of the molecule, the blue molecules, into the dorsal side, and the formation of region is called gray crescent. So gastrulation begins and starts at that location. So this gray crescent established the bilateral symmetry of the frog even before the first cleavage event, because the first cleavage event bisects. So the first cleavage bisect this gray crescent. So the site of sperm entry will become ventral to the region of the embryo, the belly, as in the figure, and the gray crescent will become the dorsal region of the frog as a back. So you can see it's another illustration of how after the entry of the sperm there is a rotation of the cortical region of the cytoplasm to appear the gray crescent. Once the gray crescent is formed, the first cleavage starts, and it must start and bisect the gray crescent, defining the dorsal and ventral axis. That's why gray crescent establish the bilateral symmetry of the frog. And hence the larva is defined from the early first cleavage to have the axis that have been said before. the effect of why the gray crescent is so important for bilateral symmetry. Experiments have shown that if we have an embryo and after the first cleavage the two blastomere are separated from each other, it will give two normal developed embryo. That's mean and equal sized. That's mean it's a kind of that if there is if the gray crescent is not important for bilateral symmetry, the two embryos arise will not be the same. Again, if we separate the two embryos when only one of the two plasmere receive the entire, so we assume that if the first cleavage does not pass into the gray crescent and we separated the two blastomeres, we'll have a normal embryo which received the gray crescent and the other just a belly piece of cells that is not differentiated. If we look again to the cleavage, you'll find that the first cleavage followed by second, third, fourth, and go on until a ball of cells is formed. And due to the concentration of the yolk at the vegetal part, there is retardation in the cleavage furrow, leads to formation of two types of blastomeres, and the cavity formed is shifted to the animal pole and is called blastocele. You can see again that the first cleavage passes through the gray crescent. Again, the cleavage furrow, of course, are numbered in orders. The second cleavage is retarded. The third cleavage is displaced toward the animal pole because the yolk is concentrated into the vegetal pole. That's why the vegetal hemisphere contain larger and fewer blastomere than the animal hemispheres. And the last one is like H, figure H represents the mid-gastrulate stage embryo. It's a transition stage between the in the gastrulate stage. If we look to real image scanning electron microscope of the cleavage, you can see how the first cleavage appear. In the below figure, it's a scanning, so you can see the first and second cleavage and the cleavage furrow, and around like the 8 or 8 to 16 blastomere stage, figure C. It's again the cleavage at the right angle. The first and second are right angle to each other. The third cleavage is equatorial and displaced, and it divides the blastomeres into micromeres, which is upper, and macromeres, which is down at the vegetal blastomeres. And this type is unequal radial holoplastic cleavage. As cleavage progresses, two populations of blastomeres arise, rapidly dividing animal micromeres and slowly dividing vegetal macromeres, and this we call it yolk-laden macromeres because they have all the yolk of the embryo. From 16 to 64 cell stage we call it morula stage, while at the 128 cell stage the blastocele is formed and it's fully formed gastrula. If you continue and see the importance of a protein that is important in adhesion, which is called the EP cadherin, while blastomeres are dividing, EP cadherin is one of the numerous cell adhesion molecules that keep the blastomere together. So this messenger RNA for that protein is already supplied by the oocyte cytoplasm. So if this message is destroyed, in other means, if the EP cadherin is not made, the adhesion between the blastomere is dramatically reduced, resulting in the destruction of the blastocele. So you can see here that control embryo, that's mean the embryo is normal. You can see the blastula with the blastomeres, while in this embryo which have the EP cadherin is depleted, you will find a massive cell with undifferentiated blastocele and no orientation and no differentiation.